Hello, my name is Thomas Mortier, and in this presentation I will briefly present some recent work on large-scale bacterial species identification with MALDI-TOF mass spectrometry data and machine learning techniques. This work is a result of a collaboration between two groups with complementary expertise at Ghent University, Belgium. In our work, we addressed the problem of bacterial species identification using MALDI-TOF mass spectrometry data and machine learning techniques. In contrast to earlier work, where traditional machine learning methods are applied on small data sets containing a limited number of species, we present benchmarking results on an unprecedented scale using data sets that contain almost 100,000 spectra and more than 1,000 different species. We compare three identification scenarios that are often not considered in literature, that is, identification of novel strains, novel biological replicates, and novel species that are not present in the training data. We use three different data sets containing labeled MALDI-TOF mass spectrometry data of different bacterial species. The BCCM LMG bacteria collection is a historical database that contains samples for more than 2,400 taxonomic reference strains, representing more than 1,000 bacterial species cultured and analyzed in the last decade by researchers in the Laboratory of Microbiology at Ghent University. Next, we used a publicly available dataset called the Lyopreservation dataset, where MALDI-TOF mass spectra were generated before and after lyophilization and subcultivation of strains. The last dataset contains MALDI-TOF mass spectra generated from isolates obtained in the recent microbial diversity project in which the bacterial and yeast fraction of a food sample was studied. For all spectra, we extracted phylogenetic information from MCBI based on the given identification label. The goal of our paper is to provide an independent evaluation of machine learning methods for bacterial species identification with MALDI-TOF mass spectrometry. To this end, we considered three different identification scenarios that are of practical relevance. In the first, most realistic scenario, we assume that novel strains of previously encountered species need to be identified at test time. In the second scenario, we assume that at test time, only novel biological replicates of strains seen during training are analyzed, and one therefore expects a better identification performance. In a third scenario, for which specific methods are required, the goal is to detect whether a MALDI-TOF mass spectrum represents a species that is not present in the training dataset. This scenario inherently translates to an auto-distribution detection task. Most of the machine learning methods described in literature, and more precisely the ones that were considered in this work, require MALDI-TOF mass spectra with a fixed length representation estimate. As such, the first pre-processing step that we considered is a so-called binning step. This step consists of dividing the mass over charge dimension in intervals, or bins, and aggregating intensity values in each obtained interval for each spectrum. For aggregation, we choose to work with the maximum of intensities, as this is frequently used in literature. The next steps in our pre-processing pipeline are baseline correction, where an estimated baseline in each spectrum is removed by using the asymmetric least squares method, and total ion normalization, where each intensity is transformed such that the sum of intensities for every spectrum sums to 1. In general, bacterial identification can be translated to a multi-class classification problem where the goal is to assign the correct class, that is, taxonomic labels such as strain, species, genus, or family to a given spectrum. Bearing in mind the availability of taxonomic information, the corresponding classification problem can be solved by using flat or hierarchical classification models. With the flat classification model, one typically denotes a model that ignores hierarchical information. In contrast, a hierarchical classification model will incorporate hierarchical information for classification purposes. In our experiments, we analyzed different classifiers which can be applied to a large-scale classification setting, such as support vector machines with a linear kernel, logistic regression, random forests, k-nearest neighbors, and a one-dimensional convolutional neural network consisting of a one-dimensional convolutional, batch normalization, and max pooling layers. In the first set of experiments, we analyzed the performance of different pre-processing pipelines and models in the novel strain scenario. In most cases, decreasing the bin size leads to a high performance, since more information is retained in the spectra. In general, most machine learning models benefit from both transformations except logistic regression, where a significant drop in performance is observed when considering total ion current normalization on top of baseline correction. A possible explanation for this result is unstable model training due to the combination of heavily reduced intensity values after total ion current normalization and specific optimizer that is used for training. In the next set of experiments, for each model, we consider a flat and hierarchical classifier and reported performance on 3 phylogenetic levels: family, genus, and species for the novel strains and novel biological replicate scenario. For all models, we consider a bin size of 5 and both transformations, since this combination gave rise to the best performance in the previous experiments. In the novel strain scenario, k-nearest neighbors outperforms the other models with an accuracy of 84.78%. On the other hand, in the novel biological replicate scenario, the outperforming model seems to be support vector machines with an accuracy of 96.15%. In both scenarios, there is no gain when considering deep learning modes, besides the fact that training is faster in case of large-scale classification settings. The same conclusion can be made with respect to the hierarchical classifiers, indicating that taxonomic information is not immediately represented in MALDI-TOF mass spectrometry data. In a final set of experiments, we analyze whether approximate Bayesian methods can be used to detect novel species during test time. Moreover, in our work, we calculate and threshold the total uncertainty in the predictions. We use a convolutional neural network with dropouts Monte Carlo and k-nearest neighbors to obtain an ensemble of size 10. By using this ensemble, the total uncertainty is approximated by means of the equation as shown in this slide. Other models were not considered due to the increasing training complexity of this scenario. When looking at the upper plots, it is clear that all the distribution samples are not clearly distinguished from in-distribution samples in the reduced input space after principal component analysis. At the other hand, when looking at the bottom plots for the reduced feature space that is learned by the convolutional network, out-of-distribution samples are clearly separated from in-distribution samples. This separation explains the better performance for the convolutional network when considering the area under the ROC and precision recall curves, as shown in the table on this slide. With this slide, I would like to conclude this presentation. If you are interested in our work, don't hesitate to check out our recently published paper. Thank you for your attention.